Hi, Mr. Poitras. So today I'm going to be speaking about neural communication, action potential, and the biochemistry behind why neurons fire. So first and foremost, it's important to note that neurons send messages electrochemically, which essentially means that neurons send electrical signals. As we already know, chemicals in the body are electrically charged, and when any chemical has an electrical charge, it's referred to as an ion. As we learned in Bio and Chem in grade 11, there are a number of important ions within the nervous system, and a couple of notable examples would be sodium and potassium, both of which have a positive charge, calcium, which has two positive charges, and chloride, which has a slight negative charge. In addition, there are a number of negatively charged protein molecules that play an equally important role within the nervous system. So it's also important to mention that neurons and nerve cells are surrounded by a membrane which allows certain ions to pass through it while simultaneously blocking the passage of others. As a result, the membrane is referred to as a semi-permeable membrane, which in scientific terms refers to a biological or synthetic polymeric membrane which allows certain molecules and ions to pass through it via osmosis. So the next thing that I want to quickly touch on is something called resting membrane potential. So when a neuron is not busy sending an electrical signal, it's considered to be at rest. And when neurons are at rest, the inside of the neuron is significantly more negative than the outside of the neuron. Although the concentrations of ions will attempt to balance out on both the inside and the outside of the membrane, this is not able to happen because the cell membrane only allows for certain ions to pass through it, while simultaneously preventing other ions from passing through what are called ion channels. At rest, for example, positively charged potassium ions are able to cross through the semi-permeable membrane quite easily. Also at rest, however, chloride ions, which are negatively charged, and positively charged sodium ions have a much more difficult time crossing through that semi-permeable membrane. Meanwhile, negatively charged protein molecules are completely unable to cross through the semi-permeable membrane of the neuron. So in addition to these very selective ion channels, there is a pump that uses energy to move 3 sodium ions out of the neuron for every 2 positively charged potassium ions that are put into the neuron. When all of these forces balance out and the difference in the voltage between the inside and outside of the nerve cell or the neuron is measured, we see something called resting membrane potential. The resting membrane potential of a neuron is approximately negative 70 millivolts, which is sometimes written as MV. And this means that the inside of the neuron is 70 millivolts less than the outside of the neuron. At rest, there are relatively more sodium ions outside of the neuron, and there are significantly more potassium ions inside the neuron. So now that that's out of the way, the next thing I want to touch on is something called action potential. So the resting membrane potential, which I was just speaking about a couple seconds ago, tells us a lot about what happens when a neuron is at rest or when it's not busy sending an electrical signal. An action potential, however, occurs when a neuron sends information down an axon away from the cell's body. Neuroscientists sometimes use words like spike and impulse when referring to these action potentials, but they refer to the same thing. Essentially, action potentials are an explosion of electrical activity that are created by a depolarizing chemical current. This means that a certain type of stimulus, such as stubbing your toe or hearing your name being called, will cause the resting potential to move towards 0 MV from negative 70 MV. When the depolarization reaches about negative 55 millivolt, a neuron will fire an action potential or an impulse or a spike. This is referred to as the neuron's threshold. If the neuron does not reach negative 55 MV, which is the critical threshold level, then no action potential is able to fire from that neuron. Also, when the threshold level is reached, an action potential of a fixed size will always fire. This is important because for any given neuron, the size of the action potential that they fire will always remain the same. In this regard, there's no such thing as a big or a small action potential within a neuron, as they're all the exact same size. So what creates these action potentials or impulses or spikes? Well, these are created when different ions cross the neuron's membrane. A stimulus, again like hearing your name being called or stubbing your toe, will cause sodium channels to open within the neuron. Because there are numerous sodium ions and more sodium ions within the neuron than there are on the outside, and also because the inside of the neuron is negatively charged relative to the outside, sodium ions will rush into the neuron. As I mentioned at the beginning of this video, sodium does have a positive electric charge, and as a result, the neuron will become much more positive and depolarized than it was before. It does take longer for potassium channels to open, but when they do, potassium rushes out of the neuron, thus reversing the depolarization. Also, at this time, sodium channels will start to close, and this causes the action potential to go back from -55 to -70 MV. And this process is referred to as repolarization. However, neurobiologists and neuroscientists more commonly refer to this process as hyperpolarization because the action potential normally goes past -70 millivolts. And this happens because the potassium channels stay open longer than they need to. Gradually, the ion concentrations will return back to the initial resting levels of the neuron, and as a result, the cell will return to its initial state at -70 millivolts. Thanks so much for listening.